Imagine that you or a loved one has a genetic disorder, a disease caused by a change or mutation in your DNA, such as cystic fibrosis, sickle cell anemia, and certain blood disorders or cancers. Now, there's often treatments that will reduce some of the pain and suffering associated with the symptoms of the disease, but these treatments will not provide a cure. Many of these conditions will get progressively worse throughout a person's life. The symptoms may get more difficult. The treatment may become less effective, and ultimately the condition may kill the person. Now consider that there is a possible cure for the disease, but it involves altering the DNA in order to correct the mutation. Would you undergo the treatment, or would you recommend that a loved one did so? In this week's Ethical Dilemma in Science, we're going to look at, should we use gene therapy to cure genetic disorders? Should genetic engineering be used to correct DNA mutations causing disease? This week's Ethical Dilemma in Science is based in part on a December 2022 article in Wired magazine taking a look at the possibility of using CRISPR, a new genetic engineering technique, to treat the descendants of an Irish king that have a hereditary condition that causes clumping of an amyloid protein around the heart muscles and nerves. Now, the symptoms start around 60, but the symptoms cause gradual deterioration and ultimately could result in chronic heart failure within 3 to 15 years. Now, there is a treatment using an RNA to interfere with the expression of the amyloid protein, but this technique requires infusion every few weeks of the RNA, and it contributes to side effects. So it isn't a cure, but it alleviates some of the pain and suffering associated with the symptoms. Now, the new approach is an idea to use this genetic engineering technique to go in and physically disable this abnormal gene, this mutated gene, effectively turning off the condition. Now, in order to understand what genetic engineering and how it affects the possible cure for the disease, it's necessary to understand what DNA and proteins are doing within your cell. Now, normally, DNA is going to be located within the nucleus of your cell, basically sequestered away in a location where it's safe and protected. Now, in order to use that information, it's necessary to make a temporary RNA copy. So this RNA copy is made from the DNA, taken out into the main portion of the cell, and it's using that information that is copied from the DNA into the RNA to make the proteins. Now, normally the proteins in the cells are going to be doing the activity of the cell. They're going to be controlling what the cell does. And so a defect or a mutation within the DNA is going to change that information. And in changing that information, it's ultimately going to change whether or not that protein is acting properly, whether or not it's going to cause a possible disease through its inactivity or through its abnormal function. Now, in order to understand what the DNA is going to be doing, we need to take a look at the DNA molecule. We basically have a two-stranded, what's referred to as a double helix because it coils around, molecule with the information coded into these little portions here that are color coordinated. These color coordinated, what are ultimately called nucleotides, are going to be copied into the messenger RNA molecule, the RNA molecule over here, and this is a single-stranded molecule that can be taken out into the cell, and by looking at these basically color-coded nucleotides here on this diagram, be able to interpret what it is that needs to be done in order to build the correct protein. So that molecule, as an artist has drawn it out based on the scientific information, is a little bit hard to interpret. And so we're going to use that information, going from DNA to a temporary copy of RNA to the functional protein, to try to visualize what's going on. And so instead of using the kind of scientific terms and the scientific nucleotides, we're going to look at it in terms of letters, because that is something that we can interpret and view. And so we're going to look at a double-stranded DNA molecule here with the information, the big dog ate the red cat, coded within it. Now, the DNA is going to be protected within the nucleus, so we're going to make a temporary copy in the form of RNA, and that's going to have that same information. And it's going to interpret that information in sequences of 3 letters. And so we end up with the big dog ate the red cat. And no, we're not going to see a protein, but we're actually going to see kind of the visualization of what that occurs. And so we've got an image from a YouTube video where we've got a dog, kind of gnawing on the head of a red cat. Now, in a mutation, what's happened is that we've changed the G into a T here, so we've highlighted it in green. And so instead, we have a big dot ate the red cat. That information is going to be copied into the RNA, and what we're going to end up with is kind of a Pac-Man situation, which is going to go through, and the big Pac-Man, or the big dot, is going to eat the red cat. Now, that is not necessarily what is supposed to happen. And so in this case, we're going to have an incorrect image of the information, and if this was a protein, this would be an incorrect protein that would possibly not be working properly. Now, to take this into a situation that is closer to reality or closer to a disease, we can take a look at the condition cystic fibrosis. Now, normally in individuals within our lungs, there is going to be a protein that is going to be involved with basically causing a dilution or movement of water into the epithelial space. Now, the protein that does that is basically moving salt ions across the boundary. In cystic fibrosis, we're missing those proteins. So we're missing the little blue things located here. And so without those proteins, what's going to happen is we're not going to move ions, we're not going to move water into this space, and mucus is going to build up. Now, cystic fibrosis is not curable. It affects many organs, but primarily affecting the lungs. This mucus buildup can impair lung function and can cause infections as this mucus accumulates blood and bacteria. And so there are a number of treatments for this. The treatment's getting to kind of clearing up the airway. So therapy vests that cause vibrations to break up the mucus. Chest physical therapy and other medicines that will go through and either open the airways or thin out the mucus so it doesn't accumulate here. But they're not providing a cure. So the idea is to provide a cure instead of a treatment for this disease. So in this case, going back to our example with the dog and the cat, we've got this genetic mutation in our DNA here. The big dot ate the red cat. That's still incorrect. But what we're going to do is move in a piece of RNA. So similar to the COVID-19 mRNA vaccines, we are going to take the RNA, put it into the cells. The information is going to have the big dog ate the red cat. And so we are going to build that protein. We are going to build that image that's going to be present there. And so even though we've got the incorrect information coming from the DNA of the cell, we've got the correct information in the form of a messenger RNA. Now, the idea of providing these cells with this temporary information for creating or building the correct protein is undergoing clinical trials right now. So 2 recent articles that have come out looking at using this inhalable messenger RNA treatment. And so using the RNA that has the information temporarily, but the correct information for building that protein can get into the cells within the lungs, and we are seeing in these clinical trials that these cells are using that information on the messenger RNA to build the correct protein. However, this is limited. The messenger RNA is only a temporary molecule. It's going to be broken down within the cell within a period of time. And so to use this treatment for the disease, we would have to continue to expose these individuals to these RNA molecules by these inhalable infusions every period of time, maybe every couple of weeks or every couple of months, in order to be able to have the benefits of this correction. Now, the goal of genetic engineering is to actually go in and correct our DNA molecule. The DNA molecule here illustrated by these words. And so we're going to use a technique called CRISPR, which is basically a form of molecular scissors that are going to go in and kind of clip out that incorrect version and replace it with the correct information. And so we're visualizing that with these molecular scissors here. They're going to go in, they're going to cut out that error They're going to cut out that change in the DNA that caused the mutation. And that DNA, once it's been cut out, is going to be removed from that, and we're going to insert in that correct information. So we've got the big dog ate the red cat. We're going to add that into our DNA molecule and correct that erroneous information, so that ultimately we have now corrected, and in what is illustrated here in yellow, is that piece of DNA that has been inserted into our actual DNA molecule, so that when that DNA molecule is read, we're going to get a functioning messenger RNA molecule that is going to build the correct protein, that is going to give us the picture of what we need to represent. Now, this CRISPR technique is relatively new. It's only about 10 or 15 years old, and it is incredibly significant. This is actually the CRISPR technique resulted in the awarding of the Nobel Prize in Chemistry in 2020 to the people that discovered it. Emmanuel Charpentier and Jennifer Doudna received the Nobel Prize in Chemistry for this CRISPR technique. And this is exciting because this is the first time that two women received the Nobel Prize in Chemistry. Now, the use of CRISPR is this incredibly strong and valuable tool for genetic engineering. But the question is this something that we should be doing? In 2023, there was actually a gene editing summit which met to talk about what types of things should be done and what types of things should not be done using this genetic engineering technique. And during that meeting, they met with a woman that underwent a cure. Basically, CRISPR was used to correct the sickle cell mutation within her body's cells. And so basically, they collected some of her body's blood stem cells, used the CRISPR technique outside of her body in the laboratory setting to correct the DNA, and then it was injected back into her bone marrow, and her cells are now producing the correct form of hemoglobin. Now, this technique has now been used in 75 people, and it ultimately could be approved for use in the United States within the next year or so. Now, these clinical trials have focused on taking cells out of the individual's body, correcting them, and then returning them. And so we're looking at what's referred to as somatic gene therapy or somatic gene editing because we're treating body cells. These individuals are not going to have their DNA changed within their eggs or ultimately their sperm. So this cannot be passed on to their offspring. And this is what this summit had. In the 2023 Gene Editing Summit, they said that, with, proper controls and oversight, it's appropriate to use this for treating body cells, but at this time, it should not be used for the treatment that could potentially be hereditable, that could be passed on to offspring. Still, there were a number of concerns raised at this summit, including the price for this technique. It could be upwards of $1 million or more for a cure in this form. So this gives rise to our ethical dilemma in science for this week. Should genetic engineering be used to correct DNA mutations causing disease? Should genetic engineering be used to treat all genetic disorders or only those that are lethal? How do we ensure equitable access to the benefits of genetic engineering, especially if a treatment could cost over $1 million? What would you need to know to make a decision about the use of genetic engineering for yourself or for a loved one? Thank you for viewing this week's Ethical Dilemma in Science. Please see the description below for additional information and references. Thank you and have a great week.